My name is Joshua McClure. I am the CEO of MaxWell Biosciences, which we fondly refer to as the Peptoid company now. I started about 4 years ago on the data, diving deep into the data. My last company was a company called Real Massive, which was a commercial real estate, still is a commercial real estate big data company. And calculating on the data is something that I've been doing my whole career. I come from a background in the United States Air Force intelligence community, and calculating based on sort of incomplete data is called mosaic theory. And what mosaic theory allows us to do is calculate on data and come to probabilistic conclusions that lead us to previously perhaps obscured conclusions. What we're doing now with the company is we are synthesizing the functional properties of peptides in a form called a peptoid. So a peptoid is when you take the side chains of a peptide and you disconnect them from the carbon on the backbone of a peptide, and you attach it to the nitrogen of a peptide backbone. So a little background on that. So if you Google this or go on Wikipedia, you'll see, carbon-based life is basically proteins and peptides with side chains that are connected to the carbon, right? Carbon-based. So our form of proteins are which we call peptoids, sort of peptide-like, right? Are these sort of medium chain molecules that are attached to the nitrogen on the backbone, and this makes them sort of uncleavable to protease. Well, the function of protease in the body is to break down proteins and to break down peptides. And so our peptoids don't break down with protease, which means you have an indefinite shelf life inside of the body, and they can be used then as drugs. So they're wasted out of the body just like a drug, right? Out the urine or whatever. So this lends a potentially new technology platform to drug development. And my particular interest is really in undoing aging, which is the conference that we're at right now. So as far as science technology in the future, I think that a peptoid structure is somewhat similar to the revolution in computing between bits and bytes, right? So bits are digital. They're either on or off, which is comparable to a small molecule. A small molecule either turns on a receptor or turns off a receptor. With a peptoid structure, it's kind of similar to bytes, right? Where you have a more data inside of the packet. And so therefore with more data, you're able to sort of exponentially increase the functional mimicry and the functional outcomes in vivo inside the body. The Barron Lab at Stanford University is really the key innovator within this field, and we've partnered with the Barron Lab at Stanford University to start to look at the antimicrobial nature of peptides, and then functional mimicry of those antimicrobial peptides inside of a peptoid structure, which is a novel and non-obvious, so therefore patentable structure. And we have a granted patent then on these antimicrobial peptoids. That's interesting. So like I mentioned at the conference, when you create a drug that has an indefinite shelf life in the body, obviously you need to look for side effects, right? And so you essentially start with many different drug candidates, and you model those computationally, but then really it's just trial and error, right? I said the other day to one of my colleagues here at the conference that we're sort of metaphorically monkeys not trying to mimic human technology, but monkeys trying to mimic alien technology, like advanced magical technology that we don't truly understand. So biotech really today is in the sort of infantile stages, where the way we test drugs is really trial and error. Did it work or not? Were there side effects or not, right? So Interestingly, your question about whether we would combine this drug with something else, right? Really, all drugs are combined with something else. So if you take a drug and you just lay in bed and you don't eat good food and you don't exercise and you don't hydrate yourself, that drug is probably not going to have the desired impact, right? Because you're not doing other healthy things. So our company is a public benefit company. So what we're trying to do is undo aging. We're trying to maximize wellness. So part of the company's formation initially in my mind was Maxwell's equations, which are the sort of the rules of electromagnetism. And at the nano level, the nano level is where the peptides are. And gravity as a force is so weak at that level that it might as well be inconsequential. And so it's really Maxwell's equations that determine all functions of peptides. And so the functions of peptides then are pretty simple. When you think about it, it's just Maxwell's equations that need to be calculated into a model to figure out what your potential drug candidates are at that level, right? So that kind of was the progenitor of the company's name's Maxwell, but it also lends itself pretty well to maximum wellness, right? So that's also sort of philosophically where the company comes from, is maximum wellness, of trying to make sure that a patient is able to perhaps get off of a medicinal drug if they don't need it. So, which I think is probably a little contrary to some of the pharmaceutical companies today, where they're just sort of treating disease instead of curing it. On the antimicrobial side, antimicrobials, the most common antimicrobials that we're used to today, which is in the very early stages of this of antimicrobial technology, right? In fact, the current stage of antimicrobial technology, which is antibiotics, which are generally produced by fungus, right? So these it's usually mycotoxins that are used as antibiotics. So it's what fungus use to kill bacteria in the constant war between fungus and bacteria, right? So those drugs are now becoming obsolete. We all have probably read articles about drug-resistant bacteria. So those drugs are becoming obsolete because the bacteria are rapidly evolving resistance using horizontal transfer of DNA to understand what kills them and how to evolve against what kills them. What's interesting about our antimicrobial peptide that we are simulating with our Peptoid technology platform, we've decided to functionally mimic a peptide called human cathelicidin antimicrobial peptide, and it's also called LL37. And I'll refer to it as LL37 from here forward. So LL37, really fascinating. So it doesn't just kill bacteria, it also kills viruses, fungus, parasites, and cancer. So it's called human cathelicidin antimicrobial peptide. The human part of it there is similar to the opposable thumb, where it is something that is actually uniquely human. So we only have one. And that cathelicidin, like Catholic Church, and acide, like homicide, is it's everything kills. It kills everything, right? So it's really interesting where we now have a peptide that we know kills everything, and it is properly evolved to be inside the human system, and has been for, prior to Homo sapiens. Hundreds of millions of years of almost no bacterial evolution to this peptide, right? So it's sort of an ideal superdrug if you could get the peptide to stay in the body long enough. So many companies have lived and died based on that idea. They died because this peptide, it dissolves very quickly inside of the body. And so our Peptoid technology platform allows that peptide to stay in the body for an indefinite period of time. So we reformulate that peptide by attaching the side chains to the nitrogen, so that the protease can no longer cleave it. Protease are like molecular scissors, and it's like taking a pair of scissors and trying to cut up the pavement. It's just not going to happen. The pavement is stronger than the scissors, right? So it's the same thing with our Peptoid system. So we have some of the function then mimicked within the Peptoid of LL37, which kills everything, including cancer and many other things. This is very well studied in LL37. Not our drug, but in LL37, right? So now what we're doing is we're trying to see, okay, what functions of LL37 have we successfully mimicked with our peptoid structure, right? What's interesting about the peptoid technology platform, the reason why it's a platform, is because we can functionally mimic, theoretically, any peptide. And there are many thousands of different peptides that are expressed by the human genome at any given time. So proving the peptoid thesis really is what we're trying to do over the next 12 months or so, is if we can prove the safety, and the efficacy, and the commercial viability of this peptoid technology platform by functionally mimicking the antimicrobial properties of LL37, and we're starting with with chronic supportive otitis media, which is a orphan disease infection of the inner ear, the middle ear, which is the mucosal epithelial layer. Remember I said that LL37 is involved in homeostasis, and it helps build that epithelial layer, especially in the mucosa. So this is a sort of natural place to test our peptoid mimic of LL37. And it's also sort of a triple or quadruple stacked accelerated pipeline within the FDA because it's pediatrics, it's an orphan disease, it's a drug-resistant strain of bacteria, which is Staph aureus. And it is suffered on a massive scale globally, mostly in the developing world right now, but it is starting to come into the developed world. So World Health Organization and CDC, NIH have prioritized it. So this allows us to very quickly determine whether or not we've met those three criteria, which is safety, efficacy, and financial viability of that drug. And we don't have to go all the way inside the body. It's still on the outside of the blood system, which is inside that inner ear area. That gets us quickly to the clinic. And so if we can do that, and the commercial pharmaceutical system then sees that we have this commercial viability, then we get a very large license payment, and that gives us the money that we're allowed to use then for all kinds of longevity and maximum wellness into the future. Our goal is really to extend sort of a natural human lifespan to around 120 to 150 years. So not super revolutionary like 200 or 500 years, but really extending it out to what if microbes were not able to attack us? What if we had such a sustainable and wholesome antimicrobial therapy that we sort of transcend a detrimental microbiome, right? Where we're optimizing our microbiome. That's right. So extending out to 150 years, what does that do, right? So that's extending a lifespan out to 150 years. So you think of a sort of a productive human lifespan, right? It's not just when we die, right? Right now, human lifespan is sort of, you're a child and you don't have really productive, you're not really contributing to society. Society is contributing to you, educating you and investing in you. And then you have this 30 to 40 year career span where you contribute back to society, right? Well, imagine if we could just extend that, so 20 to 30 years, right? Now you're adding on to the end of that, right? And not only that, but actually contributing to cognitive performance by removing the mind fog, the brain fog of microbial infection, directly impacting cognitive impairment, age-related immunodementias or age-related immunodysfunctions, which really are dysregulated production of LL37. Remember, we only have one of these antimicrobial peptides, right? And so immunosenescence really hinges all around LL37. LL37 is produced by all of the immune cells, including natural killer cells, every cell that you can think of. It's also involved in every stage of stem cell differentiation throughout the immune cell system, right? So back to what happens if we could extend out to 120 to 150 years. So generally, people retire around age 60 to 65, right? So 65, extending that out to 150. So perhaps you would die at 120 to 150, but not die gradually, like what you see in Alzheimer's, right? What if you just died because you didn't have any stem cells left, right? Your bone marrow just stopped producing. And this is all sort of without any other innovation happening, right? So if what types of undoing aging, what types of longevity and biotech innovations are we going to have over the next 40 to 50 years? Probably revolutionary in other ways, not just antimicrobial, right? So I think what our company is doing is sort of setting the table. We're sort of setting the table for other innovations to come along, right? In multiple ways. One, removing the detrimental impact, and then two, extending the productivity of these innovative scientists, where they're able to continue to be innovative during that time, right? So a good metaphor that Aubrey de Grey actually had, who organized this Undoing Aging conference, is, I've got a little bit of a beard here, very short, so do you, Adam. So you've seen Aubrey's, right? I mean, it comes down to his, to his belt buckle, right? And every time I see him, I say, I get beard envy every time I see you. And he said, well, there's a pretty easy way to get a beard like mine. And I'm like, oh, an easy way to get a beard like yours. And he said, yeah, just don't cut it, right? So that metaphor, you take it back to antimicrobial impact on longevity. What if we can get rid of the thing that is cutting our longevity, right? These detrimental microbes of all kinds, all the different kinds that even if you take antibiotics right now, it essentially, it leaves just little pockets of that microbe throughout your body. And those little pockets of microbes now are resistant to that antibiotic, increasingly so. The more you take that antibiotic, even if it's you take penicillin for one thing, and then you get an infection of another thing, and you take penicillin again, that little pocket now became more resistant to penicillin, right? So the first thing you took penicillin for now is gradually becoming more and more resistant inside you to penicillin. So inside of you, you have your own personalized microbiome that is more or less resistant to antibiotics than someone else, right? At some point, I'd like to get into how LL37 has not developed resistance within bacteria over the past hundreds of millions of years, and that has to do with a concept called flocculation. So flocculation is really interesting. It's actually used in water treatment. And flocculation, what it does is it actually, it entangles the insides of the bacteria. So your natural process inside of the human body, LL37, very elegantly just sort of spirals in through the membrane of the bacteria. So remember, bacteria are cellular organisms, right? And so imagine this bacteria, right? The LL37 is a peptide, so it's very small, sort of like on a scale, X-Wing fighter versus the Death Star, right? And so, think about Luke Skywalker zipping in through that little port, right, inside the Death Star, and then he's moving around and he's doing things. That's what LL37 does. But imagine that if the entire X-Wing fighter went into the core of the Death Star and just entangled it. So it couldn't do anything anymore. It didn't blow it up, it just entangled it. And not only that, but thousands of those things came in and just entangled, just gooed it all up, and it can't move anymore. That's what LL37 does. And this makes it so that the bacteria can no longer transfer the DNA out, right? So Cipro and other antibiotics, what they do is they pop the bacteria and it explodes out. And as it's starting to explode, as it's being attacked by Cipro and these other antibiotics, it's gathering information about Cipro, and it's writing data to its own DNA and saying, I'm being attacked by Cipro and this is what it's trying to do to me right now. And then when it explodes, that SOS message is encoded in the DNA and it's just floating around in the extracellular environment, right? And what's interesting is actually just in 2018, there's a very interesting study about that actually showed it videoed, just like what you're seeing here on YouTube or a blog or something. It videotaped a bacteria extending a pilus like an arm, almost like a blind man feeling around, because it heard that pop, heard the bacteria pop, and it reached out and it grabs the DNA of the other bacteria and pulls it in. So I'm using these words so you can Google it, right? So the DNA extends this pilus out, and it actually has, it looks like a hand on the outside, right? So it comes out and it feels around itself, looking for that DNA. It pulls the DNA inside of itself, and then reads the DNA and looks at that SOS message and says, oh, this is what just happened, and takes that DNA and puts it inside of itself, so that the next time Cipro comes around, it has the same resistance as if it was that past DNA that just got attacked. It was really fascinating, right? So genetic memory transferred horizontally as opposed to a parent-child relationship. Yeah, so with the baby boomer demographic bulge approaching us, we have an existential economic demographic problem, right? Because you can't just materialize younger people to be in their productive earning years in order to pay for that aging group, right? And so if we want to avoid an existential economic crisis, if we could extend the productivity of these older people so that they're paying for their own living, right? What this requires is two things: not just medical innovation, but sustainable, affordable innovation, right? Because the demographic bulge is not just a few people. It's not just the people who have medical insurance. It's that whole bulge that you have to take care of. It's actually the people without insurance that are most damaging economically, right? Because the government has to pay for that. And so the load gets spread out over the entire populace for that group that doesn't have insurance, right? And so if we can extend their health span, if we can make them healthier, that is the goal of the company. This is why we incorporated as a public benefit corporation, was to create health for the world safely and affordably. That's actually our benefit statement, is to create health for the world safely and affordably. And so if we can create affordable drugs that help with that problem, that it hits multiple needs of the whole global society. The most impactful research I've seen within the last 5 years, which is almost exactly 5 years actually as of now, is in 2014 and 2015. There's a group of researchers in the Bay Area in San Francisco who were researching heterochronic mouse parabiosis. And so heterochronic mouse parabiosis really became quite popular in the 1960s. And then the Nixon administration came into play, and the Nixon administration, as we all know, was somewhat over-conservative, and really put the kibosh on any kind of sort of unnatural experimentation, which means innovative, any kind of an innovative regenerative medicine approaches within the biotech community, especially in seminal research of, well, what happens if they're right? So, and it was really in the 1960s that heterochronic mouse parabiosis showed for the first time that an old mouse that was sutured to a young mouse, that old mouse would just get younger in every way, in every observable way. The biological processes, the biological age of the old mouse became indistinguishable from the young mouse. And if you disconnected the suture, that, quote, old mouse, the chronologically old mouse, would live to the same chronological age as the young mouse. It would just continue living as if it hadn't been old before, which is sort of mind-boggling in the implications if we could do that for humans, right? It would essentially mean that you would create a biologically immortal being. You could still get hit by a bus and die. A traumatic injury would still affect you, but your productive years could essentially be extended indefinitely. So I first got involved with biotech when I saw those results, and I said, there's a big data problem. So when you compare the proteins, the thousands of proteins in an old organism versus the thousands of proteins in a young organism, you have an interactome, right, which is happening in each being, but then you're comparing that as well. And the amount of computation necessary for that, I would say, is probably not even available in the entire cloud that we have today. All the computation that exists on the planet would not be able to tell you exactly what's happening there, right? Because it's dynamic. It's not a balance sheet where it's just a snapshot, right? It's constantly changing all the time. The proteins are folding in different ways at any given time, and so any individual protein has a very large amount of interactivity that it creates, right? So it's a very, very complex problem, and those are the problems that really attract me. I love these extremely large data set problems, and that's what brought me out of commercial real estate into biotech, was to look at this problem. So the most impactful research that I've seen within the last 5 years is the heterochronic mouse parabiosis experiments, which in the modern time really were pioneered by Michael and Irina Conboy, who are now at University of California Berkeley, but they did their initial research at Stanford, and then Valeda and Weiss-Correa and others. There's a really nice YouTube TED presentation by Tony Weiss-Correa on heterochronic mouse parabiosis that somebody who's not familiar with it should probably watch. But what it said is that a mammal's health span can not only be extended indefinitely, but also maximum wellness can be achieved by proteins and peptides and probably RNA and other things that are in the plasma, expressed in the extracellular environment. And this is what motivated me really to come from the commercial real estate, big data, sort of institutional side of big data into the biotech side of biotech, and start to look at can I be a part of this longevity revolution that is happening, which I think will have a really unpredictable outcomes, but I think could be extremely beneficial. All in all, I'm an optimist when it comes to that. I think that we'll see a new era of humankind coming, where we don't have to fear death so much. We don't have to worry about time being scarce, where we can invest in ourselves for long term, for perhaps 100, 200, 300-year spans of knowledge acquisition and productivity. I think to optimize technology, to optimize the application of technology, one first must be an optimist. You have to counter the fear of the unknown, right? And whenever a new technology comes out, there's always the, what are sort of euphemistically referred to as the troglodytes. The people, very conservative people who are afraid of the implications of the new technology. And you can see that in the legislation that came out in the early days of stem cells, right? Now stem cells are being used almost routinely without any kind of side effect that you would see in a synthetic drug. So I think it's important to first be optimistic, and then fund the optimists. Find the optimistic scientists who are looking at creating optimistic outcomes, optimistic futures for humanity, and look for holistic therapies that mimic the functions of the body, that allow the body to repair itself. This is self-serving what I'm saying, right? Because peptoids mimic the functions of the peptides that exist in the body, as opposed to turning off receptors in the body, right? So small molecules either turn on or turn off artificially receptors in the body. And so you're blocking the body's function, as opposed to helping the body's function and helping the body get back into a state of optimal regulation. So I think that's a sort of long-winded answer to how do you optimize the benefits of biotechnology and this biotechnological revolution that we see happening right now. Life has been described as a dance of proteins, right? And so peptides are proteins that are just short chain. They're short chain proteins, right? So if you can mimic life, if you can leverage life, and you can optimize life, the implications are pretty exciting, right? If you can start to get into this crazy advanced technology that we're born with, and get in there and start to optimize it just a little bit as it degrades, right? As it degrades, and you can just track what degraded, right? By looking at the differences between young and old plasma, and you can say, look, this is degrading over time. What if we just give it a little boost and make it not degrade over time, right? So this is in the very beginnings, the infantile stages of understanding this technology, this biological technology. It's even in the word there, right? It's a technology. If it's a technology, if biology is a technology that we're born with, it sort of almost makes us into space machines. We are space machines. We're walking around on a rock that is floating through space, and we're just gifted with this technology that allows us to resist radiation, to resist all of the other competitive life forms that are trying to eat us at any given time. The clear ones are the lions, but the invisible ones are the microbes, and they're just constantly eating you. There's a war happening inside of you all the time, right? So if we can counter that, we've done a pretty good job with the lions. I don't see any lions here in Berlin, right? So we've done a pretty good job with the lions. If we can do a pretty good job with the bacteria, it's sort of the technological and civilization equivalent of keeping the lions out, right? So you can get a lot more work done when you're not trying to be eaten, right? So that's I think where our company is headed. That's why we've decided to focus now on microbes, antimicrobial technology through these peptoids. It's a platform that allows us to launch into so many different areas that impact maximum wellness and definitely longevity. That's interesting. So you asked me if I had a 2-minute talk with a billionaire in an elevator, what would I tell them to convince them to fund my venture? So I actually have this chance. I have this opportunity on a fairly routine basis. So several of the people who are currently funding the company are billionaires, and it's because I've had this opportunity several times. And the way that I've sort of optimized that opportunity to be in the elevator with that billionaire more frequently is to health coach billionaires. So personally, on the side, let me backstep a little bit. If you were a billionaire, who would be the optimal health coach for you if you could afford it? A highly innovative biotech CEO who has connections with all of the researchers, right? And so I've had a couple of opportunities now to health coach billionaires, and almost universally, they end up investing in the company, right? And so this is something that helps with the company in a big way, and it's actually transformed the way I raise money, is because that personal interaction with this billionaire then makes them see, one, the personal improvements that they see in their health, right? When I connect them with Mayo Clinic physicians and Stanford medical technology and things where now they're getting access to things they didn't even know existed, much less getting access to it, right? But then when they see the innovative things we're doing with our company, and a lot of times in connection with the people that we just connected them with, that's when they can fund us at even at levels that are routine and simple for them, right? It's like $1 million for some of these people it's less than buying a pizza. They still think of it as a lot of money. $1 million is a lot of money, but they also see it as something that they trust will be used well, and so I can't think of a better technology than this peptoid technology to sort of prove a case that there is a new technology that is about to emerge in humanity that allows a programmatic interface to model the functions of a peptide, which is all of life, right? It's to mimic life. If you can mimic life and create programmable nitrogen-based life, this is what we're doing. We're doing it right now. So if we can prove this case, we have potentially the most valuable company in the world.